Hello everyone, welcome to the course Practical Microbiology for the second stage anesthesia department. The fifth pre-lab will be about the biochemical test and specifically about the hemolysis on blood agar. That means it's mostly related to Streptococcus species. We'll start with an introduction of Streptococcus. Streptococcus, when it's a gram-positive bacteria. When we apply the gram stain technique, it's a gram-positive and its spherical shape typically grow in chain or in pair due to incomplete cell division. We have their shape or arrangement as chain. Many streptococci are part of normal flora of human and animal, but some species can cause disease ranging from mild to severe acute or chronic infection. Very ما streptococcus بهربتر أو نورمال فلورايا يعني بشكل طبيعي سنوف جسمي مدايا هاي بس بومرو فيجي هروسا وجناريج بس هندك شوانا نسوخيو تشيتكنش خاصة ستريبتوكوكاس بايوجين نيومونيا جالاكشيا أبت هني باثوجينيكن so their pathogenicity start from mild to severe acute or chronic infection احتمال حاد بت احتمال مزمن بت حسب infection منك. So, what are the characteristics of streptococci? Microbiologically, we have many characteristics of streptococci. Gram-positive bacteria, as we said previously, they are arranged in chain or pair. Usually, they are capsulated. Not all streptococci are capsulated, but most of them are capsulated, or usually they are capsulated. They are non-motile. They don't have flagella. Non-spore forming. They cannot produce spore. They are fastidious. They need nutrients. We have they are catalyzed negative, and from this we can differentiate if they are gram-positive bacteria from the Staphylococcus genus. So Streptococcus are catalyzed negative, while Staphylococcus are catalyzed positive, and this is the shape of the Streptococcus bacteria, which is arranged in chain or maybe in pair, forming diplococci like this. If we classify the streptococcus, we have more than one classification of streptococci. This is according to the three criteria. According to the oxygen requirement, they are anaerobic or aerobics or facultatively anaerobic. Some of them are aerobic, some are anaerobic, other are facultative anaerobic. يعني هندك شونا هواينا هندك شونا لا هواينا يعني حش أكسجين ناكن وبإجار ناتشب أكسجين هابت وندك شوانجك اختيارينا يعني أكسجين هابت نابتا بخو يعني جينا خبريفت. We have according to the serology we have Lancet classification and this is mostly we will give many information theoretically. And also we have according to the third criteria, we will classify streptococcus according to the hemolysis on blood agar. So according to the hemolysis on blood agar, we have more than one type of streptococci. يعني الفيري تحلل بتت blood أختم بسر blood agar زرعت get or when we culture on blood agar, it can lyse the red blood cell and give a specific feature for each type or species of this bacteria. Streptococcus classification also we have based on their ability to cause hemolysis. The third point, because our topic will be about the hemolysis, so we will focus on the hemolysis on blood agar. What's the meaning of hemolysis? It's lysis of red blood cell because blood agar contain red blood cell. And those bacteria that can cause hemolysis, they lyse the red blood cells يعني RBC تحلل الدم producing toxin called hemolysin. When they lyse the red blood cell, the result of them it's hemolysin toxin. And according to the Brown, in 1919, they were divided into alpha, beta, and gamma. This is based on the hemolytic pattern. حسب hemolysis مثل نوعية hemolysis هاي ألفا ما هي بيتا وما هي جاما وهريك شونا صفاتك طيبة هي each of them have a specific characteristic you will see later. You have hemolysis on blood agar. The first classification we have alpha hemolysis. It causes partial hemolysis to RBC and act enzymatically on blood pigment leading to the green discoloration around the colony. When we have Streptococcus pneumonia and Streptococcus viridans, these two species of the Streptococcus genus, when they grow on the blood agar, they produce colony, and around this colony, they produce a green discoloration zone. So it means if we have those bacteria cultured in blood agar and appear like this, it means those bacteria may be either we have Streptococcus pneumonia or Streptococcus viridans, and both of these bacteria can cause pathogen or they are pathogenics. Beta hemolysis, the second classification on hemolysis, we have beta hemolysis. It causes complete hemolysis to red blood cell, leading to the formation of clear zone around the colony. And example of this type of Streptococcus, we have Streptococcus pyogenes and Streptococcus agalactiae. The colony of the bacteria when they grow on the blood agar, if they are Streptococcus pyogenes or agalactiae, when they appear like this, they have a clear zone of inhibition around the colony. This is the colony of the bacteria, and this is the zone of a clear zone of inhibition around the colony, like this. All these they have zone. Gamma hemolysis, the third classification of hemolysis, we have gamma. In this type, it has no effect on the red blood cell, just they grow their colony on blood agar. They are non-hemolytic streptococci. Example of them, they are Enterococcus faecalis. Enterococcus faecalis also is a species of the streptococcus species or genus, and their colony appear like this. Just we have colony without zone or clear zone or green zone around the colony. Why we use this or apply the culturing on the blood agar for streptococcus identification? We have hemolysis on blood agar give us it's a definitive test to differentiate between the streptococcus pyogenes and non-group A beta hemolytic streptococci. As we see here, we have alpha hemolysis. Partial hemolysis will occur and will give green discoloration around the colony, such as we have Streptococcus pneumonia. Beta hemolysis, they produce complete hemolysis and they produce clear zone around the colony, such as we have Streptococcus pyogenes, which is group A, according to the Lancet group, and gamma hemolysis, they do not produce any zone of inhibition, such as we have Enterococcus faecalis. Streptococcus identification, we will start if we have bacteria and we want to identify it if it is streptococci or not. We will start with the gram stain. If it's gram-positive cocci, we will then apply the catalase test. If it is catalase positive, it means it is staphylococcus, but if it is catalase negative, it means it's streptococcus. So, to differentiate between the species of streptococcus, we will apply the hemolysis on blood agar, or we will culture that bacteria on a blood agar. And on blood agar, they give us three classifications. We have alpha hemolytic, beta hemolytic, and gamma hemolytic on blood agar. And each of them have specific species that give the result. We have, after that, we have bacitracin for more accuracy. We have bacitracin susceptibility test and optogene susceptibility test. Also, we can apply on it. As we said previously, we have microscopic examination gram-positive bacteria cocci arranged in chains or pair like this. It's a pair or chain. We we have Streptococcus pneumonia. They arranged like this. A catalase test, if it's catalase positive, it will appear like this. We have bubble. If it's catalase negative, there is no bubble. The procedure of culturing on blood agar, we have, first of all, we will prepare the blood agar media. Then, the second step, we will inoculate the bacteria by streaking on the prepared blood agar. So we will streak on the prepared blood agar the bacteria that we want to identify it, and then we will incubate at 37 degrees Celsius for 18 to 24 hours. After incubation, we have to observe the presence of hemolysis. If there is alpha hemolysis, beta hemolysis, or gamma hemolysis, by observing the plate under transmitted light. So if there is a clear zone surrounding the colony, it indicates that it's beta hemolysis. We have Streptococcus cause a variety of disease. Why we need to identify Streptococcus because it causes many diseases. Some of them, as written here, we have Streptococcus pyogenes cause pharyngitis. Pharyngitis, it means the inflammation of the pharynx. We have also rheumatoid fever, skin infections such as impetigo and cellulitis. So, impetigo is a contagious bacterial skin infection forming pustulate and yellow crushy sores, and it's mostly found in children. So, and cellulitis, which is an inflammation of submucosa or it's inflammation of the skin, dermis, and subcutaneous connective tissue layers. And also we have scarlet fever. Scarlet fever also another skin infection that cause skin rash. skin rash بعرف بيجن مثلا لير أم بيجن لير وقت مثلا سكينتا هون جلدتا شتي برسكت بشك ورمتي لتهن. We have pneumonia, another infection caused by Streptococcus. We have pneumonia, which is the infection of the lung caused by Streptococcus pneumonia. This species mostly cause pneumonia. Pneumonia التهاب وقت lung دا تشد بيت أنجيل سيت مروفي تشد بيتن بحالتي أم بجن نيومونيا وكيش بكتيريا ستريبتوكوكس فين سوخية تشد كت we have Streptococcus pneumonia. نو في بخص Streptococcus pneumonia بس أفيد داس بيكي ما بحث كرين مثلا pharyngitis, rheumatoid fever, skin infection, scarlet fever. أفا كيش تشد كت كيش بكتيريا Streptococcus pyogenes تشد كت فينا سوخية. We have toxic shock syndrome. Toxin is a severe condition that can cause after streptococcal infection. This is occur if we have streptococcal infection after the condition of severe condition. They may change to toxic shock syndrome toxin. Also, what we have another infection. We have necrotizing fasciitis, a severe infection that can involve almost every organ system. So, about the rheumatoid fever, if a patient infected with the pharyngitis, a complication of pharyngitis infection may lead to the rheumatoid fever. So, rheumatoid fever is an inflammatory disease caused by autoimmune reactions to untreated or improperly treated streptococcus throat. So, it will cause sometimes it may lead to the damage in the heart valve, so it's dangerous. And this was about today pre-lab.